Today's lecture, it will be on sensation and perception, some of the basic terms that go along with that, and we will dive into vision. We will not finish the process of vision in this talk today. So here are some of the basic terms I need to be familiar with. And I would study these using flashcards. Transduction is the process of changing one form of energy into another, and that happens with all of our sensory neurons. So when it comes to special receptors in the eye, it takes light waves, which is a form of energy. And when light waves strike those rods and cones in our retina, transduction occurs. Light wave energy is transformed into action potentials, which is another form of energy. So anytime we have any sensation and perception, it involves transduction. Sensory adaptation is when you have a stimulation that remains constant. So if you put on a watch or a necklace like in the morning, you'll first notice it. But if it stays constant, that basically starts to fade into your awareness and you're not really perceiving it anymore, even though the stimulation is there. So we do have sensory adaptation, which allows us to focus our attention on what's changing in our environment. The two basic terms of sensation and perception are also important to note. Sensation is called a bottom-up process. That is, all the stimulation coming in from our environment is stimulating specialized sensory neurons in our body. That's what sensation is. So, and then that all is action potentials of the brain. The process of perception is imposing patterns and meaning on all those neural impulses being sent to the brain. So basically right now I'm talking to you in a barrage of words, and that's stimulating specialized receptors in your ears, sending messages to your brain. But because you know the English language, you are able to perceive, like, he just said that word, he just said that word. And the process of perception is putting it together in a pattern that makes sense. There are also things known as thresholds. And the absolute threshold is the smallest amount of stimulation necessary in order for us to detect it. So, if you've all had a hearing test, you know they were looking at your absolute threshold for hearing. What's the softest sound you can hear in each ear? Difference threshold is about a change in a stimulation. So if you can think about products, like the one I use typically in classes is Doritos. If they're trying to improve Doritos, they could say, okay, here's the original Dorito, what it tastes like. And then if they add more like nacho cheese powder to it, and you can detect it when they give that to you and you taste it, they've reached a different threshold. They change the amount of stimulation, and the average person can notice that. It's a lot of terms all at once in just about 3 minutes. I hope that was helpful. We're going to start talking about vision, but first we need to talk about light waves and their properties. The amplitude is how tall a light wave is, and that determines how bright a light is. The wavelength, the distance from the top of one wave to the next wave, determines the color. And if you think about light, that is one part of the overall electromagnetic spectrum, which is everywhere in the environment. So if you look at this is the electromagnetic spectrum. A visible light is only a portion of that. And it's basically the wavelengths of the electromagnetic spectrum that goes from 750 to 350 nanometers, which is a very small unit of measure. But also within the electromagnetic spectrum are microwaves, TV waves, radio waves, X-rays, in this room right now, and wherever you are right now, there are radio waves bouncing around. But because the receptors in our eye don't perceive that wavelength, we're not seeing those. If you turn on a real radio, you can pick up signals from radio stations. And so they're in the room. So you have light waves coming in, and basically these light waves pass through a series of structures. The very first structure is the cornea. That's the hard protective surface of the eye, and that starts to bend the light so we get a focused image on the back of our retina. After it passes through the cornea here, you can see that there's basically some fluid that I'm not asking you to know. But the next area is the pupil. So that is the little black circle in the center of the eye. There's just basically an opening. And there is the muscle around that. So when you look at the color of a person's eyes, that is the striated muscle that surrounds the hole. So the iris, the muscle, will control the size of the pupil. It can basically make the pupil smaller or make it bigger, depending on how much light is in the room. If there's a lot of light, obviously the pupil's going to get very small. Actually, I mean, if you ever have seen a photo where someone has red eyes, that's typically because if someone uses a flash or there's a bright light, the light waves will pass through a big pupil, bounce to the back of the eye where that's all being basically invigorated with blood, and so it hits the red in the back of the eye, and then that red bounces back into what's being recorded in the camera, the phone, whatever. Okay, so the light passes through the cornea first, then the pupil. Lens is next. This is a tough structure which is malleable, it's bendable, it's movable, and you see little muscles attached to it. So muscles will stretch it or crunch it together, and that is what focuses an image then on the back of the eye. Now, if you ever looked at an old or much older person, or even an old pet, an old dog, you'll see that sometimes that's cloudy, and that's known as a cataract. That's what that looks like. Typically, cataract surgery involves removing the human lens, actually by a needle going into the eye, and then emitting sound waves to crush the lens, vacuuming it out, and then replacing it with a human-made lens. So if you're wondering what is involved in cataract surgery, what it is, and that's just basically changing the lens in the eye. You have in the middle here more fluid. So light waves go through cornea, pupil, lens, and then strikes back here in the back of the eye, which is known as the retina. Now this retina has several properties to it. There are specialized cells in the back of the retina known as rods and cones. We have actually a lot more rods than we have cones. So we have about 120 million rods and about 6 million cones. And the strange thing is, if you've heard of these specialized cells, cones allow us to see in color and also in detail. But the reason, and even though we have fewer of them, they are concentrated in this structure here called the fovea. And the fovea is just loaded with cones. You can see it's directly at the back of the eye. So whenever we stare directly at something, the cones are being stimulated. So that's why we typically think we see in color all day long and in detail all day long. The rods are more scattered all around the periphery of the retina. And they allow us to see at night and in black and white. One other thing that occurs is, I'm going to show you another picture here. This is, if you can see this little black square right here, they're taking that square and blowing it up to the right. So they're taking just a little cross section of the retina, and you can see the rods and cones are back here. And then they basically send their axons forward to the axon, you have the axon terminals, which then land on the dendrites, or right up next to the dendrites of two other series of cells, bipolar cells and ganglion cells. And basically the messages are being sent deeper into the eye. Those bipolar cells and ganglion cells are transparent, so light passes through them. And the ganglion cells have all their axons leave the eye at the very back. And I'm using the cursor here to point to it. That forms the optic nerve. Where the optic nerve leaves the back of the eye, you can't have rods and cones there. It's a point of exit of the physical structure of that nerve. It's a whole bundle of axons. And so we each have a blind spot in each eye. But because we have two eyes, basically you're having information being made up for by the other eye. And we're constantly moving our eyes around, which also helps us to basically not perceive a blind spot in our eye. Okay, so those are the major structures of the eye. Okay, I'm going to spend a little time here talking then next. Oh, here's where the optic nerve leaves the eye, the fovea, where the cones are concentrated. Again, this is another picture basically of how light comes into the eye and strikes it and then stimulates those rods and cones. The last thing I want to talk about in today's recorded lecture is about color vision. We have two theories that work together to help us to understand how we see in color. The first one is called the trichromatic theory. And tri, like tricycles, like three wheels, tri means three. And chroma means color. So basically the trichromatic theory says that we have three different types of cones that allow us to see red, green, and blue light. and they're maximally sensitive to those particular wavelengths. So if you look at this, you can see, again, going with the nanometers, measuring the wavelengths of the electromagnetic spectrum, you have some cones that are maximally sensitive to the blue light, which is roughly then about 450 nanometers. And then, but there's overlap too. So they'll respond a little bit less as you get over to the 530 nanometers. They'll respond less, but then the green cones pick up and are responding a lot. And then you also have the red cones, which maximally sensitive about yellow, 600 nanometers, but they're the only ones responding when you get around the 700 level. So the trichromatic theory says we have 3 different basically types of cones that are maximally sensitive to those 3 kinds of lights. And then depending on which cones are firing, we get a mix and blend of colors that we can see. The other theory is a lot more difficult to explain, but I will try to use a brief example to illustration in this lecture to help you see it. It's called the opponent process theory. And basically this happens deeper in the retina at the level of the ganglion cells. If you can consider, and basically what it says is that these neurons have at these ganglion cells, when they're not being stimulated much at all, they have a firing rate that's like this. It's a regular firing rate. And then basically these ganglion cells, if they're stimulated with yellow, some of them will speed up and fire a lot more. And then if you take away the yellow light, they will try to return to their base rate of firing and overshoot it and then end up going slower. And so that's when you get basically an after image that you can see. So what I'm going to show you next is just a brief demonstration I'd like you to participate in. Staring at the dot in the center of this screen here for about 15 seconds. And then this is stimulating the green, the black, and the yellow ganglion cells. And each of these colors then has an opponent color. And so if you're staring at this screen for a good 15 seconds here, I'm going to flip the screen, and what I want you to do is blink real fast when you do this. And when I do that, I mean, it just happened for me, you're seeing red, white, and blue, because those are the opponent colors of what we were just staring at. So if you look at, again, we had yellow, that becomes blue. And we saw green, that becomes red. We had black, that becomes white. And the opponent process helps us understand these after images that we see. And with these 2 theories together helps us understand how we see color at all. So starting up in the next video, we'll be talking about depth perception with vision, and we'll also be talking about the process of hearing.